Thank you for the introduction as well as the opportunity to present ChemPartner's capability regarding antibody discovery using B Cell cloning approach. And thank you everyone for listening in on this talk. Today I'm going to present ChemPartner's B Cell cloning capability using Berkeley Light's Beacon platform for generating unique and potent antibodies. We wanted to evaluate the accelerated discovery of anti-PD-L1 antibodies by B cell cloning method and traditional hybridoma method. So we recombinantly expressed selected antibodies discovered by both methods and further characterized these antibodies for parameters such as binding, blocking, and affinity. So we have several different options when selecting the antibody discovery approach to use. There is either phage display, hybridoma, or B cell cloning, and there are pros and cons of each of these methods. Particularly for phage display, it can be a shorter duration of 4 months. There is flexibility in terms of types of antigens that you can use, and having sequence information early in the drug discovery process. The screening format tends to be scFv as well as Fab. But one of the challenges is that you may need to affinity mature your lead candidates. But generally, there is a large library required in order to identify really good binders. For the other two technologies, hybridoma and B cell cloning, tend to leverage the animal immunization. And the benefits of that are to have in vivo affinity maturation. So there is really no need for affinity maturation similar to what you might encounter in phage display. But with hybridoma in particular, the duration can be a bit longer, and the members you wish to evaluate could be much lower than B cell cloning method. Because of animal immunization, both methods could have challenges if it's a toxic or highly conserved targets. Generally, there has been more success with challenging targets though. The advantage is that screening format is IgG for both methods. But what we really feel is transformative with B cell cloning, especially on Beacon platform, is that you are able to really screen deeper into immune repertoire. In addition to possibly skipping overall affinity maturation process, that great sampling of immune repertoire really enables accelerated discovery as well as evaluating what the sequences are early in the process. And what's most important is being able to move function forward in the drug discovery process. So at ChemPartner, we have Berkeley Light's Beacon platform in order to accelerate antibody discovery. So on the left there is the Beacon instrument, and it is able to accommodate up to 4 OptoSelect chips, which are shown in the middle of the slide here. The OptoSelect chip actually houses a sub-nanoliter chambers called nanopens, and we use controlled patterns of light to move the single B cell into the nanopens. And within the chip, you can evaluate tens of thousands of single B cells. You can culture these B cells, allowing them to secrete antibodies, and then assay for target-specific binding as well as function, all directly on the platform. At ChemPartner, we are uniquely positioned to be able to generate the reagents needed for an antibody discovery campaigns. So for our proof of concept study using hybridoma and B cell cloning method, we generated reagents for immunization such as a human PD-L1 ECT. We generated recombinant cell lines such as human PD-L1 overexpressed on CHOK1 as well as cyno-PD-L1 on CHOK1. We also generated recombinant antibodies that were then characterized through a variety of assays. So there is a assay development group in our Shanghai facility who developed binding assay as well as the functional assays, including T cell activation assay, PD-1, PD-L1 blocking assay, mixed lymphocyte reaction assay to evaluate the functional properties. We compared hybridoma technology in our Shanghai facility to the B cell cloning capability at our South San Francisco location. We did VHVL sequencing by NGS at Shanghai facility and also did further characterization afterwards. So this is a snapshot of B cell antibody discovery workflow. The immunizations we tend to outsource or work with client to have immunizations done, but we receive the lymphoid tissue such as bone marrows, spleens, or lymph nodes from the immunized animals. And we do the isolations and plasma B cell enrichment at our site at South San Francisco. We load those B cells on the beacon and can execute binding, blocking assays or other functional assays, and then export the B cells that have the desired binding or blocking activity. Once those B cells are exported, we prepare the cDNA, amplify VHVL, and send it out for sequencing. And upon sequence analysis, we can then clone, express, and purify at our Shanghai facility and do functional characterization of these antibodies. So this is the general overview of any project to be executed on Beacon platform. It comprises of 5 different stages. Stage 1 is reagent generation, which takes up to 3 to 4 weeks, and it includes reagent generation at our location in Shanghai, or even a client can provide us the desired reagents. We perform the quality assessment of these reagents. We develop the assay for titer analysis. We also screen all the parameters on Beacon beforehand. We also optimize the post-export workflow if we are using, for example, any transgenic mice such as alloy. Stage 2 is immunization, and as I mentioned before, we do outsource this process at a local vendor. We perform a serum titer analysis, and typically this process can be for 30 to 60 days. Stage 3 is Beacon experiments, where we are screening thousands of plasma B cells on Beacon on a 11K chips, which basically allows us to screen up to 11,000 single B cells on a single day. Now we can screen 2 to 3 times more in the same week if we have excess of plasma B cells available. We typically culture those excess B cells in a proprietary mouse B cell medium. On a stage 4, we typically do the export and post-export workflow of Beacon. The export process is a overnight process and is generally automated. The cDNA generation and VHVL amplification and sequencing takes up to 3 to 4 weeks. The stage 5, where the production and characterization of the recombinant antibodies is done, it very well depends on the client's preference, because some clients prefer to have it done at ChemPartner, where our Shanghai's team would help us out in producing the antibodies as per client's preference, or some client prefer to do that at their own location. The typical process for us would be for around 4 to 8 weeks. So really what we find as having the most value for this platform is being able to move function forward in the drug discovery process. So on the Beacon chip, you're able to assay for target-specific binding as well as functional properties within the channel or within the pen. And in the next couple of slides, I will show you some of the examples of assays that we have developed on Beacon at ChemPartner. And some of the other assays we are considering on this platform include cytokine release, epitope binning, and cytotoxicity. So besides being able to screen thousands of single B cells while maintaining diverse repertoire, we feel that the most value for the platform is in identifying candidates up front that have the desired functional properties, which overall accelerates drug discovery process. So here I have shown some of the in-channel binding assays where you can see either beads or cells that express target of your interest are loaded into the channels such that a B cell that is secreting target-specific antibody will show up as a bloom with a signal right above the pen of that specific B cell. So you can potentially assay for, in a format such as this, is antigen specificity as well as cross-reactive binding. We have been able to look in a multiplex format a cross-reactive binding to mouse, human, and cyno proteins all at once. So on this slide, I have shown some of the in-pen binding assays. On left side, you can see either beads or cells that express target of your interest are loaded into the nanopen along with the single B cell. The binding signal will be detected surrounding the cell or bead if that B cell is secreting antibody specific to the target. For the blocking activity, we imported detection antibody here conjugated to elixafluorophore 488 so that we could detect the presence of secreted antibody. Then we introduced soluble ligand fluorescently labeled here shown as PE, so we can see signal in both channel suggests that the secreted antibody is specific to the target but does not outcompete ligand. But in the image above, you can see an example of blocking antibody where you see signal correlating to the antibody binding to the target, but there is limited signal in PE channel, suggesting that antibody prevents the binding of ligand to the target on either cell or bead. Now on the right side, you can see that we have labeled two cell lines expressing target of your interest with two different dyes, here green or violet. Then introduce detection antibody fluorescently labeled here with PE. The binding signal observed with two channels, PE and FITC, suggests that the antibody is specific to the cell line which was labeled green and not violet. So you can potentially assay for, in a format such as this, is antigen specificity to one target with counter screening against another, as well as cross-reactive binding. Here I have shown an example of in-pen reporter gene assay, which we have developed in early 2020. On left side, you can see a reporter cells were imported along with single B cell. We incubated them on chip for about 20 hours to detect the signal of the gene induction. If the B cell secretes specific antibody, which thereby activates a pathway, here we tested NF-kappa B pathway, which induces a signal associated with GFP reporter gene, then we will notice reporter cells turning green on beacon. The beacon instrument allowed us to capture image every hour during this incubation process. So on top, you can see images shown with interval of 4 hours, and we were able to detect the onset of signal as early as 4 hours, which thereby increases its intensity till 16 hours and then saturates. On the bottom, you can see the video with increase in the signal throughout those 20 hours. So for this proof of concept project with Berkeley Lights, we actually wanted to compare the output of B Cell Cloning campaign on Beacon platform compared to standard hybridoma approach. We had binding and functional assays as well as affinity and diversity assays done in order to compare the properties of the antibodies coming from both methods. We use identical reagents for immunizations by IP, and similar assays were used during screening process. We ended up harvesting spleen and bone marrow from the immunized animals for both hybridoma as well as B cell cloning. For hybridoma, we use SJL as well as Palpsy strain of mice, and the detection methods are a little bit different between Beacon versus screening for hybridoma supernatant. So I will go into next couple of slides describing that. Our target here is of course PD-L1, and we were able to use benchmark antibodies to evaluate our antibodies coming from these two campaigns compared to benchmarks. So comparing the B cell cloning workflow to the standard hybridoma workflow, the amount of time that it takes to get the sequences, not including the time needed for the animal immunizations, is shown here. For B cell cloning, once we have the purified B cells, we were able to load on Beacon and export the B cells that have the desired properties and then initiate the sequencing within 2 to 4 weeks total. And comparing that to hybridoma technology, the duration is quite a bit different. The process involves fusion followed by primary and secondary screenings. In this case, we did ELISA binding as well as blocking assays. We did look at human and cyno cross-reactive binding during the primary and secondary screening stages. We had to just select 40 to 50 high priority clones for production and purification. Of course, we got the characterization information, but at that point, we are actually using all of the characterization data to select top candidates before doing the sequencing. And by not having that sequencing information, it can actually create challenges later in the event that top candidates are not as diverse in terms of sequence as perhaps you would have liked it if we would have known that the sequences earlier. This slide shows the format of the ELISA-based blocking assay that was used for hybridoma supernatant screening. And so in a classical format of a blocking assay, we coated with human PD-L1 ECD-FC and came in with anti-PD-L1 antibody from supernatant and detected that either in the presence or absence of biotinylated PD-1. Now, if there is a complex formation with biotinylated PD-1, that would indicate that it was a non-blocking antibody. And if the signal completely excluded the biotinylated PD-1, that would suggest that it was a blocking antibody. So this was an ELISA-based quantitative evaluation of blocking activity, and we cast a broad net, anywhere between 30 to 100% inhibitory activity was how we selected our hits to move forward to subcloning process during the primary screening of hybridoma supernatants. So in contrast, on Beacon platform, we used image-based method for selecting our blockers versus non-blockers. So on left, you can see we loaded the pens with CHOK1 cells overexpressing human PD-L1 protein along with a single plasma B cell and allowed to secrete antibody, and then import beads that were coated with human PD-L1. So in this format, you can look at in-channel bead-based binding as well as in-pen cell-based binding. For the blocking activity, we imported detection antibody, here conjugated to elixalafluorophore 568, so that we could detect the presence of anti-PD-L1 antibody bound to the cells or beads. Then introduced soluble ligand PD-1, fluorescently labeled here shown as elixafluorophore 88. So if we see the signal in both channels, suggest that the secreted antibody is specific to the human PD-L1 but does not outcompete soluble ligand PD-1. But in the image above, you can see an example of blocking antibody where you see a signal correlating to the antibody binding to human PD-L1, but there is limited signal in FITC channel, suggesting that antibody prevents the binding of PD-1 to PD-L1 either on cell or bead. So this is the comparison of B cell cloning versus hybridoma output. So on top there is the B cell cloning results for either cell-based or bead-based binders. In total, we evaluated around 33,000 single plasma B cells on the chips. Over 200 of them bound human PD-L1 on CHOK1 cells, and of those, 35 of them had blocking activity, and over 500 bound human PD-L1 on beads as well. So on bottom here, you can see the results of primary and secondary screenings of hybridoma supernatants, where you can see over 50 bound human PD-L1 on CHOK1 cells, and similarly on cyno PD-L1 on CHOK1 cells as well. Of those, 48 had variable degree of blocking activity. So here we identified more number of blockers from hybridoma approach compared to a B cell cloning approach. However, false positive is a definite concern during hybridoma screening. The results of the blocking assay is been shown on this slide. As you can see from the table, some of the clones which were considered as blockers during the primary screen did not maintain the same blocking activity when they were purified. So basically, during the secondary screen or once they were purified, the activity was lost. So in general, 32% of the clones actually lost this activity. So even though we had more number of blockers compared to B cell cloning approach, we can certainly say that this is a low efficiency screening method because these clones actually did not retain the inhibitory activity once they were purified and revalidated. So overall, we identify 35 blocking antibodies from B cell cloning approached on Beacon platform. We recovered unique VH and VL sequences of them, and we eventually expressed 22 antibodies after the hotspot analysis. We also purified a good set of antibodies from hybridoma approach as well, and both the purified antibodies were later characterized for binding by ELISA and by Phage, as well as a blocking by ELISA, and the affinity measurements were done. The data was also compared against the benchmark antibodies listed below, and I will be showing you some of the data in the next couple of slides. So here you can see the blocking assay done by ELISA on the purified antibodies identified by B cell cloning approach as well as hybridoma approach. The assay was done in order to understand or evaluate the inhibitory activity of these antibodies by preventing the interaction between PD-L1 and PD-1. And as you can see, 19 antibodies which were identified by Beacon platform had equivalent or better inhibitory activity compared to the 11 antibodies which were identified by the hybridoma approach. As you can see from the graphs, they have a really good blocking profile and is very similar to those of the benchmark antibodies. So here we have this isoaffinity plot to really evaluate what we ended up with these antibodies from both approaches compared to benchmark antibodies. The sensograms of benchmark and top two clones from Beacon platform are shown on the right side here. As you can see that these two antibodies look similar to benchmark antibody. a very fast on rate and slow dissociation curve. We actually had several antibodies from B Cell Cloning campaign that had single-digit nanomolar affinity or better. So overall, we can say that Beacon platform certainly gave us the really good output. So here is a summary of the antibodies that we were able to obtain from B Cell Cloning and Hybridoma approach. Overall, as you can see from the hierarchical clustering tree, that most of the antibodies were unique and distant from the benchmark, but I definitely wanted to point out that the two antibodies that had a really good affinity, which are listed in this green box, are unique as well as distant from the four benchmarks that we tested. They are also listed in the red box. And I also wanted to mention that we noticed that we had all the antibodies from B Cell Cloning approach had 100% unique sequence. Unlike the hybridoma, where sometimes the dominant clone takes over the other clones and your diversity may be lower compared to the B Cell Cloning. So this is a slide basically summarizing the evaluation of the antibodies obtained from both campaigns, B Cell Cloning and Hybridoma. So overall, we can say that there are a number of factors that needed to be considered. Certainly, the time that it takes to get these sequences, 2 to 4 months for Beacon platform in comparison with 8 to 10 months in hybridoma approach. The selection criteria is extremely important because in Beacon platform we use very clear image-based selection where we look for the negative signal for the soluble ligand in case in here in the FITC channel, compared to the hybridoma approach where still it's a quantitative ELISA, but I think the selection criteria is so much variable that you end up choosing the blockers which had the inhibitory activity with 30% to 100%, which may end up being more false positives basically. So overall, we had around 35 blockers by Beacon platform and over 40 blockers from hybridoma. But as you can see, the false positive rate for B Cell Cloning is around 20% versus 32% for hybridoma. And if you are comparing their functional activity, you can definitely see the bottom half of the table where you can see their affinity, their blocking activity, as well as their functional activity in the T cell assay. And overall, we can say that we were able to get really high quality anti-PD-L1 antibodies by Beacon platform. So at the end, what we learn from this proof of concept study is that hybridoma did give us the more number of hits or the blockers, but none were more potent than the benchmark antibodies, and affinity did not correlate as well with the potency. Also, when they were tested in the functional assay, fewer had the functional activity in an orthogonal T cell activation assay. while in comparison with the Beacon platform, it obviously accelerated the whole discovery process, but we also got the chance to have a greater sampling of a immune repertoire to identify potent blocking clones, and also, as explained earlier, 2 clones which had the subnanomolar affinity, we completely correlated with their potency. So here, just acknowledging all the folks who contributed immensely in this proof of concept study. Our team in South San Francisco, our team in Shanghai, and also Berkeley Lights team who helped us understand this instrument, as well as answered all our questions and also helped us to develop new assays which can be used in our future projects. So thank you everyone for listening to this talk, and I'm looking forward to your questions. So basically here trying to answer some of the questions that you may have. For example, the first question is, what is the relationship between immunization strategy and the hit rate? So certainly there is a correlation between the route of immunization that is done. So in our past experience, we noticed that if we do the Hawk-based immunization, then we would focus on the lymph nodes where most of the hits come from, followed by the low number of hits which come from spleen. And typical duration of the immunization is around 32 days. For the IP-based immunization, which is usually for 60 days, we would focus on spleen or bone marrow. Most of the hits are basically coming from spleen. Okay, the next question is how plasma B cells are isolated and screened on Beacon. So once the immunization is completed, we get the tissue as explained earlier, and then we basically make a single cell suspension from those tissues, and then we used a magnetic commercial magnetic kit from either from stem cell or Miltenyi, which basically have a CD138 positive selection cocktail, which basically enables us to isolate plasma B cells. And these plasma B cells are then loaded onto the B cells. Typically, we would expect to get around 300,000 to 500,000 single plasma B cells from 3 to 4 mice, and we would get certainly more from the spleen though. This is what I was talking about the lymph nodes, but usually they are more than enough to screen these cells 2 to 3 times on Beacon, because we would screen a certain population of these plasma B cells on day 1. We will keep the excess B cells in the incubator with the mouse B cells, or the mouse or human B cells that we have internally developed, and then screen those remaining cells on the subsequent days. Okay, the next question is what type of screening assays that were done on Beacon. So as earlier in the talk, I have already explained you what type of assays that are done. The most common assays that are requested are the in-channel assays. You can do the binding assay, cross-reactive assay, cross-reactive assay in a multiplex format. You can also do the cell-based assay sequentially if you have multiple cell lines to screen against. You can also do in-pen cell binding and bead-based in-channel assay combined. So there are multiple ways of doing this assay on Beacon, And we have certainly tested a couple of different configurations that have worked on number of projects that we have done in past. We have also done the in-pen binding and blocking assay, which is also seemed to be very popular, certainly because you can identify the potential candidates having that blocking activity early on. And recently our projects also been focused on these reporter assays, where we were able to identify certain clones which basically activates the pathway, and that was basically seen in form of GFP signal over a certain time. So those kinds of assays are also can be done on Beacon platform. Okay, and the next question is what is the overall recovery and diversity of the clones? The overall recovery in our experience is typically around 80%. So we have basically have, let's say, about 100 hits from Beacon. There is also an attrition rate from the identified hit up to the getting the sequences of them. At certain stage, typically our cDNA recovery is really good. It's around like 95%, but at some times you would probably lose some of the clones during the VH and VL amplification stage, as well as the sequencing stage. So typically in our experience, we were able to recover around 80% of the clones. And the good thing about is whatever clones that we were able to recover had the 100% unique pairing between VH and VL. So basically you will lose 20% of your clones, but the remaining ones were all 100% unique in terms of the sequences. Okay, and that's all the questions. Thank you everyone.